Today role of Vir genes inside Agrobacterium and plant was presented by Victor Athaziam Sri Kaliswari College Shivakasi. After wounding in dicot plants, the phenolic compounds were released. The phenolic compounds were sensed by Agrobacterium by Vir A gene. In case of monocot plants, either acetosyrangone or hydroxyacetosyrangone was act as a inducer. Once recognized, the acetosyrangone or wound vira become autophosphorylated and become activated. Phosphorylated vira along with chromosomal virulence genes G and I activated the virulence gene. Virg called as activated Vir-G. Activated Vir-G activated the Vir-Box gene subsequently. ddNA was excised from the T plasmid by VIRD1 and VIRD2 nuclease activity. VIRC1 binds to the overdrive sequences near right border. Inside Agrobacterium, virulence gene A, virulence gene G and virulence B were activated. Next, VIRD4 and type 4 secretion system and VIRD5 were also activated. A single-stranded binding protein VIRE1 and VIRE2 and VIRF also activated. They were exported into the cytoplasm. In mechanism of tDNA transfer and its integration into the genomic DNA was reported recently. Next, to bacteria. Some genes were activated in plants, and those are necessary for the tDNA integration in plants. The Vir proteins were exported into the plant cell, cytoplasmic trafficking and nuclear targeting, and tDNA integration into the host genome. The T4 secretion system export Vir proteins from Agrobacterium to the host cell. Vir2, a single-strand binding protein covalently attached to the tDNA complex and prevent from the host nuclease activity. Single-stranded binding proteins binds to the tDNA. And in the 5' the nuclear targeting protein VIRD2 was attached. Vir2 interacting proteins, that is VIP, that interact with Vir2, thereby preventing the activation of plant defense genes. The tDNA complex import an alpha-mediated pathway through the nuclear pore with the help of cellular proteins. Initially, the tDNA complex directly bind with Vir interacting protein bind with VIRE2 and helpful from plant degradation mechanism. The tDNA complex import through the nuclear pore with the help of cellular proteins. Initially, VIRD2 NLS sequence interacts with importin alpha and import into the cell nucleus. Inside the nucleus, the tDNA was randomly integrated into the host genome. Thank you.